All right, this is a tough question if you're not sure how to structure or approach it. We want to know how many cases in the genetic code would you fail to know the amino acid specified by a codon if you knew only the first two nucleotides. We know that a codon is made up of 3 individual letters. We'll just put X in here now to show that this is what a codon is. So let's say we have a G and we have a U, and we don't know what the other one is. G, U, something. If I go look at the genetic code, I know that this could be a U, a C, an A, or a G, and they would all code for valine, otherwise known as VAL. So this is an example of when we know the first 2 letters, we know exactly what the amino acid is. It doesn't matter what's in the third position here. It could be a U, a C, a A, or a G. We're going to end up with valine. So this would not be an example of what we're talking about. But what if we were looking at a codon that started with AA and then something after that? What are the options from the genetic code? Well, we could have a U, a C, an A, or a G. Now, if it's a U or a C, then we're going to end up with asparagine, which is N, or ASN. But if it was AAA or AAG, we'd end up with lysine, or K, or LYS. So in this case, if we don't know the third position, I can't tell you if it's going to be asparagine or lysine. It's going to be one of those two, but I don't know which one. So this satisfies the scenario that we've asked for and the problem here, where when you know the first 2 nucleotides bases, but we don't know the last position in the codon, that we don't know what amino acid is specified for. If you look at the genetic code and look at all the cases where this happens, there are a grand total of 8 scenarios in which this is true.